Thus far, the chapter has concerned itself with the chromosomes that are always present in pairs in higher organisms, so-called autosomes. However, there's one set that is not necessarily present in a perfect pair, and those are the sex chromosomes. And so we number the other chromosomes in humans. They're numbered 1 through 22, and we call them autosomes. But the sex chromosomes we call X and Y. And in humans, as well as in many insects, flowering plants, etc., the female has 2 X chromosomes. We call this situation homogametic. While the male has an X chromosome and a Y chromosome, and we call this situation heterogametic. What happens to these chromosomes during meiosis? Well, females always produce gametes with an X chromosome, while males produce 50% gametes with an X chromosome, and they produce 50% gametes with a Y chromosome. And as you can imagine perhaps, if there is a trait whose gene is on the X chromosome, this can make inheritance of those traits a little more complicated. And there aren't many genes on the Y chromosome, certainly not in humans, and so we're going to go ahead and ignore that one for now. But when a gene is on the X chromosome, we call that trait X-linked. And X-linked traits have a few defining characteristics. First, males are never heterozygous for an X-linked trait, right? They only have one X chromosome, so they only have one allele. Females can be heterozygous because they have 2 X chromosomes. And that means that these traits show up in different ratios in males and in females. Heterozygous females transmit each of those alleles to half of their offspring, and a female can receive a recessive allele but still show the dominant trait, right? However, a male who inherits an X-linked recessive trait from their mother always exhibits the recessive trait, right? Because there's not a second X chromosome in their cells for them to be heterozygous, right? They only have that one trait, they're always going to exhibit it. And that means that X-linked traits show up much, much, much more frequently in males. Let's consider a concrete example of this, which is the disease Fragile X syndrome. Fragile X is a recessive X-linked genetic disease caused by a triplet repeat expansion in the FMR1 gene on the X chromosome. And I actually have a dear friend who is a carrier, and so she has one X chromosome with the repeat expansion, and so we'll go ahead and call that XF, and she has one X chromosome with the normal number of repeats, and we'll call that one X+. Her husband is phenotypically normal, and so her husband has an X chromosome with a normal FMR1 allele, and he has a Y chromosome. These two folks have 3 children, 2 boys and a girl. And remember, each of these 2 boys has a 50-50 chance of getting the normal allele from their mom and a 50-50 chance of getting the disease-causing allele from their mom. And so one boy received the normal allele, and of course he received a Y chromosome from his dad, and so he is phenotypically normal. The other boy received the Fragile X allele from his mom and a Y chromosome from his dad, and so he is profoundly autistic as a result. He is showing the symptoms of Fragile X. And the daughter, well, the daughter received a normal allele from her dad, and she could have received either the normal allele or the Fragile X causing allele from her mom. But because the disease is recessive, it doesn't matter, right? She is also phenotypically normal. How can you detect that a gene or a trait is sex-linked? Well, the clue is actually right there in the name, right? Transmission of a trait is different in males and in females. And in particular, reciprocal crosses give different outcomes. For example, if we consider the white-eyed trait in fruit flies, we have three different alleles, if you will. We have X plus, which is the wild type allele on the X chromosome. We have XW, which is the white-eyed allele, and we have the Y chromosome. And so a white-eyed male who is XW and Y crossed with a red-eyed homozygous female, X plus, X plus. If you cross the two of them, all of the male progeny are going to be X plus Y, which means they're going to have red eyes. And all of the female progeny are going to be X plus XW, and so they're also going to have red eyes. However, if you take a white-eyed female who is XW, XW, and you cross that white-eyed female with a red-eyed male who must be X plus Y, then all of your males are going to be white-eyed because they're XWY, and all of your females are going to be red-eyed, right, because they are XW from the female and X plus from the male, right? So the males are going to be white-eyed, the females are going to be red-eyed, but remember, these two crosses, right, this cross and this cross, these are reciprocal crosses, right? We took females with a mutant trait and crossed them with wild-type males. We took males with the mutant trait, we crossed them with wild-type females. And in this case, when the trait is sex-linked, we see different outcomes from those reciprocal crosses. One last thing. The word always, right? Always gives white-eyed males and red-eyed females, is doing kind of a lot of work here, right? And so now imagine that you're in Thomas Hunt Morgan's position, right? And you've observed, say, I don't know, 5 male F1 progeny. And of those 5 males, you're expecting to see a 1-to-1 ratio of different phenotypes, right? Red-eyed and white-eyed. But instead, all of your flies have white eyes. Did you just get unlucky? Right? If you looked at more flies, might you eventually see the ratio that you're expecting? At what point do you decide that you've looked at enough information, that you've looked at enough observations, that what you've observed is so improbable that there must be something else going on, right? At what point do you decide that your theory about, or your hypothesis about what is happening at the molecular level must be different than what you're expecting, right? As you might expect, the field of probability and statistics has quite a lot to say about this particular question. And so we're going to return to the role of probability in predicting the distribution of progeny next.